I'm Dr. Mhandowa of Asante Health, and today I'm welcoming Dr. Marco Ruggiero, a molecular biologist and medical doctor, to discuss with us his exciting development of an innovative immunotherapeutic tool named IMUNO. Hi there.
Hi. Thank you. Thank you so much for this beautiful introduction, and thank you for this great opportunity. I'm here at your service to try to explain this truly exciting development that occurred in the past couple of years or so.
Thank you. Thank you very much. It's a pleasure to have you up here with us today as well. So we're looking forward to having our discussion today. So Dr. Ruggiero, first, please tell us about your training and work experience.
Well, unfortunately, this could be very long considering how old am I. I am 63, but I'll try to make it as short as possible. In 1980, I got my MD at the University of Florence. I have a specialization in diagnostic radiology at the University of Siena, Italy, and a PhD in molecular biology at the Ministry of University in Rome, Department of University in Rome. In 82-83, I served in the Army as medical officer, and in 1984, I moved to the USA, working for Barrows Wellcome, Big Pharma, together with Dr. Eduardo Lapetina, and I had the honor to publish the paper in PNS, Proceedings of the National Academy of Sciences of the USA, sponsored by Sir John Vane, a Nobel laureate in 1982. And this paper was on protease inhibitors in the days when the AIDS epidemics just began to be recognized. In 1985, back at the University of Florence, Italy, I published a paper together with colleagues, Professor Vincenzo Chiarugi and Simonetta Vannucchi, on the supramolecular assembly of plasma heparin, the heparin that we have in our blood. And I mentioned this paper because this was the beginning of a development that eventually 33 years later led to the design and development of the supplement that will be the main topic of today's talk, IMUNO. Then in 1987, I went back to the USA, this time working for the US government at the National Cancer Institute of the NIH in Bethesda, Maryland, working with Dr. Stu Aronson and Professor Peter Duesberg from Berkeley, California. I was working in those days on the genes responsible for cancer, the so-called oncogenes and signal transduction. In 1988, I had the opportunity to publish a paper in Science on signaling related to the EGF. Today, this gene is very well known for its involvement in breast cancer. And I began to be interested in what today is called immunotherapy. We didn't call it this way in those days. And in 1990, I published together with colleagues from the NCI and NIH a paper on macrophage colony stimulating factor and human cancer. And please notice that this was about 4 years before Professor Yamamoto from Philadelphia published the first paper on the so-called GcMAF macrophage activating factor. I went back to Italy, I got tenure at the University of Florence, I became full professor of molecular biology, and in 2009 I began to be interested in what it is truly called now, and it was called then also immunotherapy. Immunotherapy, that is to harness the power of the immune system to fight diseases, mainly cancer and neurodegenerative diseases. And also I began to be interested in the role of the microbiome. Today everybody is excited about the role of the microbes in our bodies, but in 2009 it was still something very little acknowledged, that we have so many microbes in our body, we're only 10% human and so on. In 2010, I worked together with Dr. Yamamoto, and we published a study, we presented a study at the 18th International AIDS Conference in Vienna, Austria, demonstrating the effects of GcMAF, this macrophage activating factor, in human monocytes that are human macrophages. In 2013, I began collaboration with Dr. Jeff Brestreet, world authority in the field of autism. And in 2014, together with Brestreet, we published a paper on transcranial ultrasonography as a tool for diagnosis and therapy in autism in a journal called Frontiers in Human Neuroscience. 1 year later, together with Bradstreet again, just before his untimely death, we published in Frontiers in Neuroscience a paper where we hypothesized, we thought that we had found the pathogenesis of autism that is related to impaired flow of lymph from the brain through these deep cervical nodes. And this paper is now in the top 1% of all papers ever published by this journal. 2015, I moved to the USA, where I'm now. 2016, I began collaboration with another famous doctor in the field of alternative complementary medicine, Dr. Dietrich Klingert, originally from Germany, working in Seattle, Washington. And together with him, we developed a protocol that is called the Ruggiero Klingert Protocol, or RK Protocol, published in the American Journal of Immunology, where we described a novel protocol for diagnosis and treatment of chronic conditions ranging from chronic Lyme, that is the expertise of Dr. Klingert, to autism and other chronic conditions. And in 2018, based on a number of papers and years of research in this field, I invented, so to speak, or I would say I designed and developed the IMUNO, which is the supplement that will be discussed today. All in all, I have about 232 publications between peer-reviewed papers, book chapters, congresses, presentations, and so on. You can go in Google Scholar and you can find them all. You may find that I have about 3,500 citations as of yesterday. And some papers, like the one we wrote together with Bradstreet, has a great number of views for a scientific paper, of course, 27,000 views. And as a matter of fact, as I said, it is in the top 1% of all Frontiers articles. So this is a very short, of course, I will omit all the details of research, what I did, where. I think that these are the most important steps in my scientific career.
Fantastic. Thank you very much. And that brings us to the next question, actually. If you could please tell us about your current research interests, actually.
Well, you know, research is a matter of being curious, and I've always been very, very curious. When I was a student of medicine, I was fascinated by the topic of cancer. First of all, because in the mid-70s, we knew very little about cancer. We didn't know anything about genes and so on. And so it was truly a mysterious disease, but it was a disease that was different from all other diseases. So until then, medical researchers were concentrated on finding infectious agents. Cancer was something completely different. So this curiosity has never left me. And as of today, my main topic of research is still cancer, but at the light of today's achievements. So today, I would say immunotherapy of cancer as it relates to signal transduction. What does it mean? Cancer is a disease of communication. Cells don't understand each other, don't communicate well with each other, and they end up harming the host. So cancer cells, in a sense, are not enemies, are simply misguided, so to speak. And so the communication within the cancer cells, the communication between cancer cells and the cells of the rest of the body, and the communication inside the cells is truly fascinating, because the more we understand, the more we can do something, let's say, to fix the problem, to re-educate, without sending them to camps, of course, to re-educate the cancer cells so that they can go back to normal. This is something that is doable. It is called reversion of cancer phenotype. And as a matter of fact, there are a few papers that you can retrieve in PubMed demonstrating that you can revert the cancer phenotype. So you can make cancer cells become normal. One of these papers was published by a research group in 2015 in a prestigious cancer journal. It is called Anti-Cancer Research. And over there we describe the negativization, as we say, of oncogene expression. So cancer cells don't appear cancerous any longer, also at the genetic level, through immunotherapy. And of course, today I'm focused on the study of immunotherapy as a way to harness the power of the immune system to fight cancer, but also other chronic conditions. As I said before, in 2009, I began to become interested in microbiome medicine, what today is called microbiome medicine, that is how the microbes influence our well-being and how are they involved in diseases, and how are they involved in the development and function of essentially all our organs and systems, including the immune system and the brain. Together with an author from the USA, Mr. Peter Greenlaw, we published a paper entitled Your Third Brain, indicating by third brain the information that is contained in the microbiome. I was the first to coin the words brain microbiome, because until recently it was thought that the brain was sterile. It is not. We have microbes in our brains, just as we have microbes in our gut, just as we have microbes on our skin. And those microbes inside our brains are as important as neurons and glial cells. They are essential for the functioning of our brain, of our mind, the way we think, the way we function. So the words brain microbiome as a sort of a primacy was published in a paper of mine that you can retrieve in PubMed in 2016. And today I'm working on studying the way microbial cells communicate within each other and with our cells. So still I'm sort of a researcher of communication, not communication between humans through Twitter or other media, but communication between cells, cancer cells and healthy cells, microbes and our cells, microbes with microbes, microbes with viruses, and so on. I am fascinated by this because, again, I think that, as it often happens with humans, wars or fights are started because of lack of understanding, because of misunderstandings. And if this is true for humans, and it is true, it is even more true for cancer cells or microbes that out of a sudden become enemies of ours, whereas usually they're friends of ours, and so on. So communication between cells, you could say, is the main topic of today's research. In the field of autism, I'm still pursuing the role of ultrasonography that we started with Dr. Brestrit in 2013 in a diagnosis and treatment, and of course, what is the role of the immune system and the microbiome in autism and neurodegenerative, neurodevelopmental diseases. And finally, this is sort of a hobby. I am working on something that may appear loosely related to whatever I've said thus far, that is gravitational time dilation and biological quantum entanglement. These are very exotic topics. You may think that gravitational time dilation, that is when you are closer to the center of gravity, time slows down. So you may think that these are things for physicists or for people interested in astronomy. Well, this is true, of course, but it is also of interest to the biologist and the medical doctor, because we have seen that you can slow down the passing of time thanks to this idea, this hypothesis that is 100 years old by Einstein, of course, by simply introducing some gravitational attractor in the proximity of DNA. So small things, they work like big planets and stars and black holes. And biological quantum entanglement also is something very fascinating that also influences the way this supplement IMUNO works. So these are my main topics of interest, my main topics of research in these days, and I shall be more than happy to elaborate on these if these are of any interest to you.
Absolutely, fascinating. And I guess that also leads us next to what led you to develop IMUNO.
Oh, well, this is something that I would say came natural. Came natural by working in the field of immunotherapy, and came natural by working together with Dr. Yamamoto, who was the one who in 1994 first coined these words, GcMAF, macrophage activating factor, and had the idea that this way of stimulating cells of the immune system, macrophages, could lead to immunotherapy of cancer and HIV infection. We began working with him, and as I said, in Vienna, we presented our first results at the most prestigious World International AIDS Conference. And then we kept on working on GcMAF back at the University of Florence. We published in a journal that is devoted to cancer immunology and immunotherapy, and it is called Cancer Immunology Immunotherapy, where we demonstrated that this factor not only activated macrophages, but also inhibited what is called angiogenesis. Angiogenesis is that process that leads to the formation of new blood vessels. You know, when cancer grows, it needs blood and oxygen, but blood vessels aren't there, so the cancer has to build its own blood vessels, like lines of supply. And this process, of course, leads to greater cancer expansion. So if you could block this process, you deprive the cancer of its blood, its oxygen, its nutrients, and so you can stop the growth of cancer. We demonstrated that immunotherapy also worked in this sense. In 2013, still working on this factor, we were the first to propose a molecular model on how this factor interacted with cells. And then we began to realize that maybe the protein of the GcMAF was not the most important part of the molecule, but the fatty acids and the vitamin D3 that were bound in a multimolecular complex were responsible for the biological effects that we and many others were observing with this factor. And this leads back to the original idea of Yamamoto, who in 1998 thought that GcMAF could work as an adjuvant. Adjuvants are those oil-water emulsions that are used in vaccines to enhance, to improve the immune response to a given vaccine. So this was Yamamoto's idea of 1998, and we began to realize that this hydrophobic, hydrophilic, that is a sort of oil-water interaction at the cellular level, was responsible for the effects that we were observing. At about the same time, so between 2013, 2014, several papers came out because of the interest induced by this type of novel immunotherapy. So many papers confirmed what we and others had observed, but also many papers they began to question the interpretation of the results. And for example, there was a paper demonstrating that people with cancer, they have normal levels of GcMAF. So if you have a normal level of something and still you have cancer, quite evidently there is no deficiency of that factor, and quite evidently, even if you gave that factor and you observed some effects, that weren't due to some deficiency. And so these and many other inconsistencies at the molecular and biochemical level that are too boring to be mentioned today led us to rethink, or let's say reinterpret, the results that we and many others had observed. Because again, the effects of GcMAF in cancer have been independently replicated by scores of researchers, and also the effects of GcMAF and autism have been replicated. So it worked, this type of immunotherapy worked, but maybe the original interpretation that everything was due to a given protein that is called GC protein was not correct. So in 2016, we came up with the idea that we published in a journal called Medical Hypothesis, that is devoted to publish exactly hypothesis, that the active site of GcMAF was indeed not a protein, but a glycosaminoglycan that is called chondroitin sulfate. That is complexed with hydrophobic, sort of oily molecules such as oleic acid and vitamin D3. And retrospectively, this was consistent with our paper published in Anti-Cancer Research in 2014, where that is actually, and very aptly I would say today, entitled Oleic Acid, Deglycosylated Vitamin D Binding Protein, Nitric Oxide, and Molecular Triad Made Lethal to Cancer. So we didn't know at that time, but we had already focused on the hydrophobic, hydrophilic, let's say oil-water emulsion as the responsible for the clinical and biological effects. So this intuition led to the development of a supplement called Rerum that was an emulsion of chondroitin sulfate, oleic acid, and vitamin D3. But then from Rerum, where we observed excellent results, we moved to this current development that is called IMUNO, that represents a sort of a quantum leap, a big leap, because it features non-animal derived ultra-pure chondroitin sulfate. So we're not dealing with human blood derivatives, we are not dealing with animal-derived compounds at variance with GcMAF or Rerum, and ultra-pure phosphatidylcholine and D3. So we're not dealing with mixtures of oils, because we found which one of the several, say, oily molecules were responsible, and D3. So IMUNO is a sort of the culmination of more than 30 years of research. You may remember that in 1985, when I was a young postdoc at the University of Florence, we published this paper demonstrating how glycosaminoglycans are assembled in human blood. And it is from that inspiration of more than 30 years ago that through a series of successive and very logical steps, I designed and developed IMUNO. As of today, we have 3 peer-reviewed papers published on IMUNO, and a 4th one has just been submitted describing the effects of IMUNO in autism and how it is integrated in the Ruggiero Klinger protocol, and we hope that soon this paper will be accepted and published. So it's a logical sequence of events that led to IMUNO, did not come out of the blue, and it is based on more than 30 years of research.